In the world of science, few breakthroughs have generated as much excitement as gene editing, with the ability to alter the very DNA that makes us who we are. This technology promises to reshape the future of medicine, agriculture, and even the way we approach human evolution. But what exactly is gene editing, and why is it so revolutionary? Gene editing refers to the process of making precise changes to the DNA within living organisms. One of the most well-known techniques is CRISPR, which stands for clustered, regularly interspaced, short palindromic repeats. It sounds complex, but it's simply a tool that allows scientists to cut and paste DNA with remarkable precision, like editing a paragraph in a book. The CRISPR-Cas9 system was discovered in bacteria, where it functions as a defense mechanism. Bacteria use it to store snippets of viral DNA in their genome, which allows them to recognize and destroy those viruses if they return. Scientists have harnessed this natural process, adapting it to cut DNA in any organism, plants, animals, and even humans. But CRISPR is just the beginning. As we move beyond CRISPR, new techniques are emerging, each with its own potential to change the world. Let's start with the impact of CRISPR in medicine. The ability to edit genes has opened the door to treating genetic disorders like sickle cell anemia, cystic fibrosis, and Duchenne muscular dystrophy. Imagine a future where diseases caused by faulty genes can be corrected at the molecular level before symptoms even appear. The implications for improving human health are profound. Beyond curing inherited diseases, gene editing holds promise for cancer treatments. By editing immune cells to recognize and attack cancer cells more effectively, scientists are working on personalized treatments that could be more effective than current therapies. In agriculture, gene editing could revolutionize food production. By editing the genes of crops, scientists can create plants that are more resistant to pests, diseases, and harsh environmental conditions. This means higher yields and more resilient crops, essential as the global population grows and climate change puts pressure on food security. CRISPR has also been used to develop animals with improved traits, such as disease-resistant livestock, or even pigs that are better suited for organ transplantation, offering hope for reducing the shortage of donor organs. But with great power comes great responsibility. The ethical considerations of gene editing are as important as the scientific advancements. The ability to alter the human genome raises important questions. Should we edit the genes of embryos? What are the risks of creating genetic inequality? How do we balance innovation with caution? In the case of CRISPR, the debate is ongoing. While the potential to cure diseases is immense, there are concerns about unintended consequences, mutation, off-target effects, and the possibility of misuse. Looking ahead, gene editing technologies are only going to get more advanced. Beyond CRISPR, newer systems like CRISPR-Cas12, CRISPR-Cas13, and base editing offer even greater precision and fewer side effects, bringing us closer to a future where gene editing could be used to treat virtually any genetic condition. At the same time, scientists are developing ways to ensure that gene editing is safe, ethical, and accessible to all. The future of gene editing is not just about science, it's about shaping a better world for everyone. The promise of gene editing is still unfolding, and as we explore its potential, we are reminded of the incredible power that science has to transform our lives. With continued research, careful consideration, and ethical responsibility, the possibilities are boundless. To harness this power effectively, it is crucial to establish robust regulatory frameworks that balance innovation with safety. Collaborative efforts between researchers, ethicists, and policymakers will be essential to navigate the complex landscape of gene editing. Public engagement and education are also vital to ensure that society understands and supports the responsible use of these technologies. As we move forward, the ethical dimensions must remain at the forefront, guiding us toward a future where gene editing not only enhances individual health, but also promotes social equity and well-being. To achieve this delicate balance, transparent communication channels must be established to facilitate open dialogue between all stakeholders. Regular forums and workshops can provide platforms for sharing insights, addressing concerns, and refining guidelines. Additionally, incorporating diverse perspectives, including those from marginalized communities, can help in crafting inclusive policies that do not perpetuate existing inequalities. Ethical review boards should be empowered to oversee research projects, ensuring compliance with established norms and identifying potential risks early. By fostering a culture of ethical responsibility and continuous learning, we can harness the transformative potential of gene editing while safeguarding against its misuse.